Good afternoon. We are here again to start with the lymphatic system. The lymphatic system. My name is Francis Anthony. I work with St. Carol's School of Nursing, and I'm here to teach you the topics under the lymphatic system. The lymphatic system is the system that augments the circulatory system. When fluids move out of the capillaries into the tissue spaces, sometimes it's difficult for them to come back and join the capillaries at the end of their work. So the lymphatic system, they will suck the fluid, filter it, and then add it to the circulatory system. So that's how the lymphatic system works. The lymphatic system have got lots of organs. We have the spleen, we have the thymus, we have the lymph nodes. And their function is to filter the lymph. They filter the lymph. And interestingly, the lymphatic system houses the immune system. They house the immune system. So we will see how exactly they work. So here, we are looking at the lymph nodes, the thymus, the spleen, the large intestines, the appendix. They all have lymphatic organs in them, especially the areas around the groin and then the armpits. They have lots of lymphatic vessels and lymphatic organs in those areas. The tonsils is at the back of the throat and at the back of the mouth in the throat, so that anything that tries to enter the body that is disease causing will be dealt with there. Then we have the thymus, which is for the maturation of the T lymphocytes. Then we have the spleen, which filters blood to remove old age blood cells, especially the red blood cells. Then the appendix. The appendix that is also at the beginning of the large intestines to deal with any pathogens that were able to go past all the mouth, the stomach, and then the small intestines. When they get there, they deal with them. Then we have the small intestines also having the Peyer's patches in them that is used to deal with pathogens that find themselves in the digestive system. Now, let's look at the job of the lymphatic system. The lymphatic system, which consists of vessels and organs, play two vital roles in our lives. One is that the vessels essentially maintain interstitial fluid levels by adding excess fluids as well as any plasma proteins back to the cardiovascular system. The organ houses the critical immune cells, such as the lymphocytes, which carry out the body's defense against infections and diseases, as well as offer acquired immunity. Now, the characteristics that they have: the lymph is excess tissue fluid carried by the lymphatic vessels. And the properties of the lymphatic vessels is that they work just like the veins. They return fluid in one way back to the cardiovascular system. They have no pumping station. They have no pumping station, and the lymph moves towards the heart, milking with the action of the skeletal muscles, the rhythmic contraction of smooth muscles, in the vessel walls. Lymph is usually a clear, colorless fluid similar to blood plasma, but low in protein. Its composition varies from place to place. After a meal, for example, the lymph absorbs fatty acids and glycerol, and that makes it look milky. But after that, when you find lymph in other places, it's just like how water looks like. Just that it's a bit thicker than water because of the plasma proteins that they contain. Lymph may contain viruses, bacteria, cellular parts, and even traveling cancer cells. So that is why they have a filtering unit. They filter it before they add it to the blood. Now, what you see on the board is edema or edema. The Americans say edema. The British says edema. And it is a collection of excess fluid in tissue spaces. So what you can see here, the arms are swollen, the fingers are swollen. If you put your finger into it, it will remain a depression on the surface of the skin. With this lady cannot wear any jewelry. Jewelry that has to be worn must not be, I mean, she can't wear tight jewelry. No rings, she can't wear it. And so you have to be careful if you have a patient who has that condition. In this case, you will not be able to find a blood vessel if you are to give an injection into a vein. You can't see anything because it covers all the blood vessels. Okay. Now, the interesting thing is that edema has no cure. So you have to manage it. Okay? If it's in the feet, you raise the legs when you are seated. If it's in the feet, you have to make sure that your legs are raised when you are seated. You massage that part of the body that has got the edema. Massage it, and then you make sure that you lift it. It's just to try to return the lymph back to the circulatory system. Anything that causes increased capillary pressure, such as decreased plasma proteins, increased capillary permeability, or lymphatic blockage, can result in swelling and congestion of the extravascular compartment. Here in this diagram, we are looking at how lymph is collected. Okay, around the tissue spaces, there shouldn't be too much tissue fluid. Otherwise, the cells will drown. And when they drown, they will die, causing necrosis. And so, the lymph will suck all the fluid and then filter it and add it to the circulatory system at the venous end here. At the venous end here. Okay. So, the lymph capillaries, they have overlapping walls that form a mini valve-like structure, and the fluid leaks back into the lymph capillaries. The capillaries are anchored by connective tissue and by filaments. Higher pressure on the inside will close the mini valves. Now, the vessels that carry lymph, we call them lymphatics. The vessels that carry lymph, we call them lymphatics. They are thin-walled and they are similar to veins. Small lymphatics are similar to capillaries in that they are more, except that they are more porous. Larger lymphatics also have collecting vessels and they have valves in them to prevent backflow. Remember that the lymph has no pumping station like the blood. The blood has the heart pumping it, but the lymph has no pumping station. So the body has to find a way of sending lymph back, and that is by exercising the body, by raising that part of the body, especially the leg, when it is swollen. And in the day, in 24 hours, the lymph that is added to the circulatory system is about 3 liters in a day. But in a day, the blood that is pumped around the body is so much. So that is how slow the lymphatic system works. There are 2 large ducts, the right thoracic duct and the thoracic duct. They both empty into the right and the left subclavian veins. Lymph flows to the heart in only one direction, one way. And because it is a low pressure pumpless system, but it is able to move with the action of the skeletal muscles and pressure changes in the thoracic region during breathing. Okay, lymph carries harmful materials that enter the vessel, like bacteria, viruses, cancer, cellular parts, and so on. And the lymph node is a germ-filled lymph, and it filters the lymph before it is returned to the blood. We are going to look at how the lymph node looks like. The defense cells within the lymph nodes are the lymphocytes and the macrophages. The macrophages are the big eaters. They are white blood cells, and they eat bacteria and digest them. And the lymphocytes provide immune response to antigens. So the lymph vessels and then the lymph nodes, we've looked at them. They are all over the body except in the central nervous system. Except in the central nervous system. We've looked at this diagram, remember? We've looked at it. So it's just showing where the lymph nodes and then the lymphatic organs are located in the body. You won't find any in the brain. So this is a lymph node. It looks like a bean. The lymphatic system consists of 2 semi-independent parts: the lymphatic vessels and the lymphoid tissues and organs. The lymphatic system functions to transport fluids, excess fluids usually, back to the blood, and they play essential roles in body defense and resistance to diseases. The lymphatic organs, the largest lymphatic organ is the spleen. What the spleen does is that it removes, by filtering all the old age red blood cells from circulation. Remember that the red blood cells have a period of 120 days. That's their lifespan. At the end of the 120 days, because of their squeezing into small capillaries, they get through wear and tear. So at the end, they can't do the work that they are supposed to do. So the spleen will filter the blood to remove them, okay? And then dismantle it with the liver and send all the recycled part, like the iron is reused by the body, and the part that is not recycled is sent out of the body. The spleen destroys red blood cells, and it acts as a reservoir of blood. It is the largest lymphatic organ, and it does that by filtering the blood of bacteria and antigen-filled cells. The next largest is the thymus gland. It produces the hormone thymosin, and it functions in programming lymphocytes, T and B cells. The T cells mature in the thymus gland. The tonsils, they are smaller than the thymus gland, and they trap bacteria and other microbes in the throat. The Peyer's patches, they capture and destroy bacteria in the intestines, and therefore preventing them from penetrating the intestinal walls. The lymph node is kidney-shaped. It looks like a bean. It can be very, very small. It can be large. And the outermost layer is known as the cortex, and it contains a collection of lymphocytes. The innermost layer is called the medulla. It contains phagocytic macrophages. So this is the diagram I wanted us to be aware of. This is the diagram that you must be able to draw and label this diagram. The lymphatic vessels contain the lymph nodes, and the lymph node is an example that you see up on the board. You have the capsule, which is the outermost covering. The capsule is the outermost covering, and the next layer is the cortex, and the innermost layer is the medulla. And it has a lot of afferent lymphatic vessels that enter, and only a few or one or two efferent vessels, because it needs to take time to filter, to remove the material that can harm the body. So fluids enter the afferent vessel, and after they have been filtered, they exit from the efferent vessels. The germinal center of follicles, they contain the B cells, the plasma Bs, and the medulla contain the phagocytes that they are used to eat the bacteria and other pathogens that can cause harm to the body. The spleen, as we said earlier, filters blood of bacteria, viruses, and other debris. They destroy worn out blood cells. It then returns some of the breakdown products of the red blood cells to the liver for recycling, example, iron, and the red blood cells, that the part that cannot be used, they are flushed out of the body either through the urine or added to the digestive substances so that it can come out with the feces. The unusable portion of the worn out blood is excreted sometimes also in the bile. Another function of the spleen is to store platelets and act as a blood reservoir. Lymphocytes are produced, red blood cells are also made here during the fetal period. So, the body's defense system is the immune system. What we just described is the lymphatic system. Now we are moving on to the immune system. We have the physical and the chemical barriers. The physical barrier is an intact skin, a skin that is not broken. If the skin is broken, pathogens can pass through and cause harm to the body. And the skin has got a large flora and fauna. Okay, there are so many bacteria living on the surface of the skin. We also have sweat. The skin produces sweat that destroy some of these pathogens, and also sebum, which is the oily secretions coming from the skin. They also destroy the pathogens. Mucous membranes are found on the inside of the hollow tubular organs, especially in the digestive tract, in the lungs. Tears contain lysozyme that destroy pathogens that try to enter the eye. So in this diagram, you can see that the skin, okay, is made up of several layers, and so they prevent the disease-causing organisms from entering or gaining entrance into the skin. Then we are looking at mucus. Mucus is lining all the tubular organs so that they protect the layers below them. If any pathogens tries to enter, the mucus traps them, and the cilia, which is located on the free surfaces of the epithelium, beats backwards and send it out of the body, either through coughing out or coughing and then swallowing, so it passes out with the digestive tract. Tears have got lysozyme in them, and if any bacteria or pathogen tries to enter the eye, the tears destroy it. So the immune system responds to inflammatory reactions and also immune response. We have the white blood cells. They are the soldiers of the body. And the white blood cells are continuously made in the bone marrow. We also call them the leukocytes. Leuko means white, and cyte means cells. So the white cells. There are a variety of leukocytes, and they are all coming from the hematopoietic stem cells. And the leukocyte that is specific for immunity, we call them lymphocytes. Lymphocytes. So here, I want us to look at this. The lymphocytes are coming from here, okay? The lymphoid progenitor. We have the T suppressor, C cytotoxic, T helper, CD4, CD8, and so on and so forth. We have monocytes on the other side, monocyte, dendritic, mast cells, basophils, eosinophils, platelets, and then neutrophils. They fight in a different way. They have a different way of fighting. But the lymphocytes, they have a specific way of fighting, and they are very good in their fighting. We call them competent. They are very competent in the way they fight. So, your immune system, my immune system, has got levels of immunity. The first line of defense, we are looking at the physical barriers. The first line of defense, we are looking at the physical barriers. The skin, which is the outermost protective covering that we have. The mucous membranes and the cilia. Inflammation, we are looking at innate response and phagocytes plus other white blood cells. Inflammation is an innate response. It's inner response when pathogens enter the body. Adaptive immune response are cells that have memory of past infections, and they are released to defend invaders. White blood cells, they are also coming from the bone marrow. There are three groups of them. We have the macrophages, the big eater, the big eaters. They are phagocytic cells. When we say phago means eat, okay? And macro means big. So macrophages, they are the big eaters, and they are phagocytic in action. Two types of lymphocytes are found here, the T lymphocytes and then the B lymphocytes. The T lymphocytes and the B lymphocytes. T stands for thymus, and B stands for bone marrow. Now, innate and acquired immune response. We have two lines that are non-specific. The skin, the mucous membranes, the secretions from the skin, and the mucous membranes. They are the first line of defense. They can get rid of anything that they encounter. The second line of defense, we call it the phagocytic cells, like the neutrophils, the antimicrobial proteins, and inflammatory response. The third line of defense, we are looking at the lymphocytes, the antibodies, and then the macrophages. We are going to look at them one by one. How does bacteria get into the body? If the skin is intact, it's very, very difficult or impossible for them to get into it. But if the skin is open, then they can get into it. The mucous membranes line the hollow tubular organs, and the microbes that can go past these portals of entry. When you breathe, when you touch something, when you breathe, you touch your eyes. If your fingers are contaminated, you transfer the pathogens to your eye. You touch your genitals, you put your fingers in your mouth. You are actually ingesting microbes. Of course, if you get a splinter or a cut in the skin, then the microbes have a full day. They can enter any time. So, the pathogens, we have non-specific defense mechanism and specific defense mechanism. There are barriers that try to stop them, but the mucous lining from the respiratory, from the digestive, from the urinary tract and so on, they deal with it. And the acid secretions in the stomach also deal with pathogens that enter with swallowing. And if they are able to go past, then you are looking at inflammation. You are looking at inflammations. And phagocytosis can come in when the white blood cells are encountered by these bacteria or pathogens. And interferons can also do that. Now, for the specific immunity, we have cell-mediated immunity and antibody-mediated immunity. When we say cell-mediated, it means that the cells themselves are the ones fighting the pathogen. If it's antibody, it means that the white blood cell produces antibodies, and then the antibodies destroy the pathogens. We will look at them in detail. Now, inflammation and its response. When cells are injured, when cells are hurting, they all secrete chemicals like histamines and kinins. And these chemicals cause blood vessels to dilate and capillaries to become leaky. Now, when this happens, pain receptors become activated. So you feel pain. One of the signs that there is inflammation is that a person feels pain. Okay? And this pain that a person feels will attract other phagocytes and lymphocytes and even neutrophils to the area. The white blood cells, the soldiers will come, and it will lead to inflammation response. Three things. One is to prevent the spreading of the damaging agents to nearby tissues. So the part will be raised. The swelling will cause that part of the body to be raised so that it will not spread to other tissues. And then there is a disposal of pathogens and a state that is held for healing to take place. So this picture is showing the process that happens. For instance, somebody gets poked by a nail. The nail has got a lot of pathogens on it. So the first thing is that the pathogens enter that part of the body. And there is pain. And because there is pain, information is sent to the white blood cells, especially the neutrophils. They are everywhere. They go everywhere. The neutrophils, they pass out of the capillary wall, and then they go to the site, and then engage in positive chemotaxis. They get into positive chemotaxis, okay? Because inflammatory chemicals diffusing from the inflamed site act as the chemotactic agent. All right? So the white blood cells immediately move, especially the neutrophils. They move to the spot. The inflammatory response here is that the tissue is invaded locally. There is release of the chemical histamine. That blood vessels become leaky, they dilate, and then the white blood cell starts moving out to go and fight. There is swelling. There is swelling as a sign of inflammation. There is pain as a sign of inflammation. There is redness as a sign of inflammation. There is heat as a sign of inflammation. And the macrophages too can move to the site. Now, containment and destruction of pathogens. Fibrinogen now in the tissue clots, trapping the pathogens. And heparin prevents clotting at site of injury. And pathogens are in a fluid pocket surrounded by the clot. And chemotaxis, leukocytes are attracted to the chemotactic chemicals to the site, so that the neutrophils, which are the first to respond, they engage in phagocytosis. There is a signal for more of them to come, called respiratory burst, and secrete cytokines for recruitment of macrophages and more neutrophils to come to the site. The macrophages and T cells secrete colony-stimulating factors to stimulate leukopoiesis, so that more white blood cells will be produced from the bone marrow. After all that, the tissue that has been invaded must undergo repair, and the blood platelets and endothelial cells in injured area secrete the cytokine that stimulate the fibroblasts to multiply and synthesize collagen, facilitated by hyperemia that provides materials needed and heat that increases metabolism. Fibrin clots may provide a scaffold for repair, and pain limits that part of the body so that healing will take place. Now, the lymphocyte that is found at the infection site occur as a result of innate response, something that is within. We have the macrophages responding, neutrophils responding, dendritic cells responding, and natural killer cells also responding. The macrophages are large phagocytic cells, we've said that before, that act as scavengers, devouring pathogens and worn-out cells. The neutrophils, which are the first to respond, they destroy invaders by the use of digestive enzymes because they can engage in phagocytosis. The dendritic cells eat pathogens and activate other immune cells. And then dendritic cells take pieces of the pathogen and put it on their cell surface so that they can remember them when they see them or they come into contact with them the next time around. The natural killer cells, they kill virus-infected cells and cancerous cells. So here, we are looking at the inflammation. You can see the redness, okay? You can see the swelling. And this is to ensure that this part will not spread, the pathogens will not spread to that side. So that part of the body is raised up. And you can see white blood cells moving out of the blood capillaries to that area. And that process of white blood cells moving out of the capillaries to that area is referred to as diapedesis. Diapedesis. Okay. So, the difference between acquired immunity and innate immunity is simple. Innate is something that is within, built within. Acquired is something that you acquire when you get the disease. You acquire the way to fight that infection. So acquired immunity is antigen specific, and it is usually systemic, and it has memory. It recognizes previous encountered pathogens. Another thing that we have to discuss is the local infections and systemic infection. The local infection, the pathogen has caused an infection of tissues at the surface of the skin or a mucous membrane. But the systemic infection from the skin or the mucous membrane, the pathogens either enter the bloodstream or enter the lymphatic system. That is why we say systemic infection. Now, during systemic infection, acquired immune response will, 1, be able to recognize specific antigens. 2, make specific antibodies and killer T cells. 3, then have memory of these antibodies and T cells for a future encounter. T stands for thymus, and then B stands for bone. So the B cells they gain their maturity in the bone marrow, and the T cells gain their maturity in the thymus gland. And the T cells, they have the hormone called thymosin to help them gain their maturity. The B cells actually mature and gain their immunocompetence in the bone marrow. And the lymph nodes, in the lymph nodes, the B cells transform into plasma cells which produce antibodies. There are so many antigens. Antigens, we have self-antigens and non-self. The non-self is what is coming from outside the body. The self is the one that is inside the body. So an example of non-self, you are looking at the food that we eat. They contain non-self antigens. Pollen grains, bacteria, viruses, fungi, they are all coming from outside. So they are referred to as non-self antigens. Now, the memory that the lymphocytes have, some of the B and T cells have memory. They remember. They remember. And when they remember, they are able to use the same skill that they used to fight in the previous encounter. So, memory cells have the ability to divide on a short notice to produce more of all of the T and B cells. And this is because of acquired immunity. They have fought the battle before, so they can fight again. After the initial response, the B's and the T's, which are amplified in response to antigens, are reserved, and they circulate in the lymphatic system for years. And some of them even circulate for the rest of the person's life. If same antigen enters the body again, then the immune response will take place rapidly and without full-blown illness. Example is chickenpox. When you get chickenpox, you are immune for life. You can't get chickenpox again. Another one is measles. You get measles, the immune system remember them, so you can't have it again. Now, an APC is an antigen-presenting cell. And what the antigen presenting cell does is that a protein from the bacterium or the fungi is recognized by the body as foreign. And the APC can be dendritic cells or macrophages. And they ingest the pathogen and place pieces of the pathogen on their cell membrane so that they can trigger a response from T cells. I want us to watch a short video and then we will continue from there. I just want you to see how the T cells and the B cells fight.
But you get sick. Here's him and Moby. How do we get sick? From... Cut it out. No one likes to have a fever or a cough that just won't stop. But you get sick a lot more often if it weren't for your immune system. The immune system is your body's way to fight off illness and disease. It finds invading cells and disables their power to make you sick. Immune system cells are all over the place, but they do tend to concentrate in your thymus, lymph nodes, bone marrow, spleen, tonsils, adenoids, lungs, liver, and intestines. Oh yeah, and especially in your blood. You can think of your immune system as an army of little warrior white blood cells that are ready to attack invaders. Tiny organisms of bacteria, viruses, and fungus try to get into your body because it's a warm place to live with plenty of food. These invaders are called pathogens, and they are your immune system's enemies. Your immune system is made up of two types of white blood cells, T cells and B cells. When pathogens enter your body, they are detected by your immune system. T cells spring into action to coordinate the immune system's counterattack. B cells analyze the invading pathogens and build proteins to fight them off. Your immune system sends lots of germs running for their lives on a daily basis. But sometimes your immune system is just not quick enough and you get sick in the time that it takes your immune system to reach full strength. When you get a virus like the flu or a cold, your body can usually knock it out on its own. Bacterial infections like strep throat may require assistance from antibiotic medications. Well, actually, your immune system does need your help. Simple things like not getting enough sleep or not eating right can weaken your immune system so that you get sick more often. You may have heard of a virus called AIDS. AIDS stands for Acquired Immune Deficiency Syndrome. The AIDS virus attacks T cells and weakens your immune system to the point that your body can't fight off pathogens. When this happens, something as simple as a cold can make a person really sick. Researchers are learning more every day about how to fight the AIDS virus, but it's still real serious. Oh, the coolest thing about your immune system is that it remembers. When you have something like chickenpox, your body memorizes the antibodies it built to fight it off. If that chickenpox pathogen tries to enter your body again, the immune system will know how to get rid of it. That's how things like polio and measles shots work. Your body is injected with a tiny amount of the virus. Not enough to make you sick, but enough to put your immune system on alert. If those diseases ever do come your way, they won't make it very far. Once you have the antibodies for a certain illness, you're immune to it. Well, the common cold is actually lots of different viruses that produce similar symptoms. Just because your immune system fought off one cold virus doesn't mean it'll be ready for the next one.
Right. So from the video, we saw that the B cells, they manufacture antibodies, and they use antibodies in fighting. But the T cells are the ones that see the pathogens and then they spring into action, causing the others to also get ready so that they can fight and fight well. So acquired immune response is where antibodies are formed. And the antibodies are formed by the B cells, and they are what is used to destroy the pathogens so that that process is known as the antibody-mediated immunity. Now, the major histocompatibility complex proteins, they are proteins called the HLAs, or human leukocyte antigens, and they are expressed from genes located on the chromosome. And there are two classes, major histocompatibility 1 and major histocompatibility 2. The one has the surface or the antigen recognized by CD8 receptors or on killer T cells. The MHC2, these are surface proteins that are recognized by or they bind to CD4 receptors, and we call them helper T cells. So we've talked about B cells, we've talked about T cells. But T cells, we've talked about killer T and helper T. Killer T and helper T cells. The killer T cells are the ones that use, the cells themselves engage in fighting. But the B cells, they produce antibodies, and the antibodies are what is used in the fight. Now, acquired immune response, we are looking at 4 phases. Recognition of the pathogen. That is when they see that, that is the pathogen coming in. Then they get the others. Everyone come. The disease-causing organism is here. They get the others. Everyone comes around. And then if there are not many, they clone themselves. They divide themselves. And then they attack. Okay? And when everything is done, they slow down. So that is RAS. Recognition of invading pathogen, amplification or the clonal selection, and attack, and then slow down. So, phase 1, recognition. We are taking it one by one. The dendritic and macrophages are drawn to the injury site, and they have consumed pathogens and presented their antigens on outside of their cell surface already. And this is read by the helper T cells. The helper T cells are the ones that blow the alarm. The antigen is a marker on the surface of a foreign substance that immune system cells recognize as non-self and trigger the immune response. The helper T cells, also sometimes referred to as the CD4 cells, which activate killer T's, CD8, and activate B cells and their maturation to plasma cells. And the plasma cells are the active form of B cells, and they make specific antibodies. The role of the helper T cells is that the helper T cells respond to the needs of the antigen-presenting cells, and the dendritic cells detect antigen and act as antigen-presenting cells. Remember, they've met before. And the helper Ts can then trigger the clonal selection of the T killers and help the T also to trigger clonal selection of B cells. And clonal selection is amplification. When they clone themselves, that means they have become many, then they can fight well and win the battle. Okay? So here in the diagram, you can see the macrophage, and that is the microbe or the antigen. So when it engulfs it, it puts a bit of the shape of the antigen on its surface. That is it. That tiny, tiny, small things. The tiny, tiny, small triangles on their surface. So that when the helper T binds with it, it forms the memory. They recognize them. It forms a recognition for them. Okay? And this is the antigen presenting cell, or APC. The APC. Now, the T helper cells are known as sometimes the CD4, and the killer are known as CD8. The CD8. The helper T multiply rapidly and trigger the production of killer T cells and the B cells in the spleen and lymph nodes. The cytokines, the chemical messengers secreted by the lymphocytes, help regulate and coordinate the immune response, and that is the interleukins and the interferons. Now, amplification. The T helper cells multiply rapidly and trigger the production of the T killer cells and the B cells in the spleen and in the lymph nodes. And the cytokines, chemical messengers secreted by the lymphocytes, help regulate and coordinate the immune response with the interleukins and interferons. Now, the attack. Phase 3 is attack. When they have multiplied, they have cloned themselves, they can all attack. The killer T will strike at foreign cells and body cells that have been invaded by infected antigen-presenting cells, identifying them by the antigen displayed on the cell surface. And they puncture the cells to sacrifice. The macrophages will clean up. So here, the cells are the ones fighting. The cells are fighting themselves. And so, the B cells will do it differently. They are stimulated to multiply by the helper T cells. They produce large quantities of antibodies. And remember in the small video, the short video clip that we saw, the antibodies are shaped like a Y. The antibodies are the ones that they use to fight. They throw that at the enemy, and it was like a bomb or a grenade of some sort. They throw it at the enemy, and there's an explosion, and that's it. Okay, and because the B cells are using antibodies, their way of fighting is known as antibody immune response or antibody mediated. Now, the phase 4 is when the danger is over. So the T suppressor cell halts the immune response by secreting chemicals that suppress the T and B cells. The dead cells in the fight are filtered by the lymphatic system. That is the spleen, and then they are excreted. So, incubation, this is when the virus and the bacteria that cause disease are multiplying. You don't have any symptoms yet, but you are very weak. Your immune system has recognized the enemy and is at stage 2 or stage 3, and you are very contagious. Another B and T cell difference is that the antibodies can only affect the outside of cell directly, or they must use indirect means, others such as macrophages, to get inside the cell. That is if the microbe is inside. The killer T's, they lyse the cell to get the infected cell. They punch holes in them. So we have talked about B plasma, B memory. The plasma cells are the ones that will make the antibodies. The memory cells are the ones that remember. Then the T cells, we are looking at T helper cells. They are the ones that blow the alarm, causing the others to come together and clone themselves. Then we have the T killer that engage in fighting itself. And then we have the T suppressor. When the fight is over, the suppressor T will tell everybody or secrete chemicals that will help everybody to stop the fight. Otherwise, they will continue fighting. And when they fight and fight and fight, it will lead to autoimmune diseases. Autoimmune diseases. So that when somebody gets an infection for the first time, these lymphocytes will have to learn how to fight. They fight the infection, and then they put a little bit of the antigen on their surface or the pathogen on their surface so that the next time they come, they remember them. Okay? And if there is a secondary infection, then the memory cells are there. They remember them. So they are not able to, they are able to gather themselves quickly and then fight. So the response is not too severe. Okay? They are able to work fast so that within a short time, they have done the work. So the two types of humoral immune response is what we are looking at. We have the antigen, and it's found on the surface. When there's a primary response, they remembered it. So they have the antigen, a piece of it on their surface, so that they will be kept in the circulation. When they divide, they will be kept in the circulation. All right? Then, if there is a secondary response, they know it already. So they can rally themselves in no time at all, so they can fight. And then the B cells differentiate into memory cells and plasma cells. So they are at the bottom of the diagram. You can see the plasma cells, then you can see the memory cells. The memory cells, they remember the antigen or the pathogen. And then the plasma cells, they make the antibodies so that they can use it to fight. All right? Okay. So we are moving on. The antibodies that are manufactured by the B cells, we call them immunoglobulins. We call them immunoglobulins. They are soluble proteins secreted by the B cells, the plasma Bs, and they are carried in the blood plasma, and they are capable of binding specifically to an antigen. They are capable of binding specifically to an antigen, and the antibody matches the antigen that is on the surface of the plasma cells. So the antigen-antibody complex, we are looking at the formation of the antigen-antibody complex. And there is one, neutralization. Virus or bacteria can't attract due to antibodies that have been bound on their surface receptors. And there is precipitation and agglutination, the formation of insoluble complexes due to the binding of antibodies, antigen determinants. That is the precipitation, which also form bridges and causes clamping of the cells, which we call its agglutination. Basically, the microbes are immobilized. The microbes are immobilized. So that is it. Now, the B cells maturation occur in the bone marrow, but in the lymph node by a maturation process that is caused by the T helper cells. And the B cells will form or transform into plasma cells so they can produce antibodies. And the plasma produce antibodies. Antibodies are what is also called the immunoglobulins. There are 5 of them: immunoglobulin M, A, D, G, and E. We call it in short MAG. And antibodies of each class have slightly different roles. And this is coming from the plasma Bs. They have slightly different roles. The immunoglobulin M can fix complements. A can be found in mucous. D is important for activation of B cells. G can cross the placental barrier, and E is involved in allergies. Now, when we come in terms of classes, antibody A is a monomer in the plasma, a dimer in the mucus, saliva, tears, milk, intestinal secretions. In fact, all body secretions containing immunoglobulin A. All body secretions, sweat, saliva, they all contain immunoglobulin A. Immunoglobulin D is a monomer and the B cell membrane antigen receptor. Immunoglobulin E is also a monomer on mast cells, and it stimulates the release of histamines that attract eosinophils immediately to bring about hypersensitivity reactions. Immunoglobulin A is the most abundant of them and is the only one that crosses the placental barrier so that it will give immunity to the fetus that is coming out into the world. The fetus, the baby, when it's born, is so innocent. So the mother must be able to give it some fighting skills, and that is immunoglobulin G. And immunoglobulin M is a pentamer, and 10% in the plasma, 1 degree immune response, agglutination, and complement fixation. Now, the natural killers, they kill by bringing out certain chemicals, and we call the certain chemicals paraffins, so that this paraffin is selected at bad cells so that they destroy the cell. Okay? And the target cell's membrane, as a consequence, will disintegrate. When they secrete the chemicals, they dissolve the cell membrane. And when the cell membrane is dissolved, the cell is killed or destroyed. There are other complements, 20 proteins by themselves, and they are innate. However, when attached to a pathogen, when they are attached or they are fixed to a pathogen or an antigen, they become activated, and then they attack the membrane of complexes, and these produce lesions and holes, lesions or holes in foreign cells and surfaces, cell surfaces. The membrane attack complexes, these are also known as MAC, and the MAC produce lesions or holes in foreign cells, surfaces, and amplify inflammatory response. The next thing we want to talk about is vaccines. Vaccine development is based on acquired immunity. We have two types that confer active immunity and passive immunity. Weakened cells, when the weakened microbes are given as injection to an individual, it's as if the person is fighting the disease for the first time. So the immune system recognizes it the next time the real disease comes into the body. And so the immune system will build itself. That is active immunity. So when we were growing, we were given injections, polio, vaccinations, measles, mumps, rubella. We were given all those vaccinations so that our bodies can fight when the real thing comes along. Passive immunity, it gives just the antibodies. It doesn't give the weakened or the organism or half the organism. It gives just the antibodies. In this case, a person has already been exposed to the pathogens and needs antibodies fast, or a person is traveling abroad and doesn't have time to wait for the time lag period for antibodies to build. Doctors give patient a shot of gamma globulins, which is a mixed bag of antibodies which will last for a couple of weeks and then disintegrate. When you are traveling, you need to go for shots, okay? And by the time you are back, the system would have adapted, because basically no country wants to import diseases to their country. They don't want that. So when you are traveling, you are supposed to obey the protocols. Okay, so this is a table between active immunity and passive immunity. Active immunity, we have naturally acquired and then artificially acquired. Active immunity, we have contact with the pathogen. You actually get the disease. Active immunity. Passive immunity, where you get the antibodies passed to you, like a mother giving breast milk to the child. The child will have to get some of the protection from the mother before he can be, as he grows, okay? Then, artificially acquired, you are looking at vaccination, dead or weakened pathogens, and artificially passive, you are looking at injection like gamma globulins. So your B cells encounter antigens and they produce the antibodies. And the active immunity can be naturally or artificially acquired. The naturally acquired is when you get a disease like chickenpox. So you build the immunity. But if you haven't had chickenpox and you want to go for chickenpox shot, that will be vaccination, and that will be passive immunity. All right? Not natural. It's artificially passive. Okay. We are talking about allergies. Sometimes you eat certain foods and you have itches all over your body. That is allergies. And allergies are hypersensitivities of the immune system. They are normal. They are not normal. Allergens are what will prompt an allergic response. Histamines are released by mast cells in an allergic response. Histamine causes blood vessels to dilate and they become leaky, causing runny nose and mucus production. Mast cells They are cells that are produced in the bone marrow but migrate to and are found mostly in connective tissues. Mast cells play a key role in inflammatory process. When activated, a mast cell rapidly releases its characteristic granules, and various hormonal mediators like histamine and others are put into the interstitium, and mast cells can be stimulated to degranulate by direct injury. So that is a picture of the mast cell, okay, binding to immunoglobulin E. Remember, that is for hypersensitivity, and the granules release histamine and others, brings an allergic attack, and common allergens are animal dander, pollen, dust, and so on and so forth. Now, sensitization. This is when you are initially exposed to an allergen and the time for your B cells to produce antibodies with immunoglobulin E against it. You have no symptoms at this time. However, as the immunoglobulin E will bind to the mast cells, the next time you are exposed, you will have an allergic symptoms as allergen that will bind to immunoglobulin E, and mast cells will release histamines and other chemicals. It is these chemicals that induce the symptoms of allergic response. Allergic response. Okay, passive immunity. This is showing a mother breastfeeding her baby. And the mother's milk, the first milk that comes out of the nipple, is known as colostrum. Colostrum. The mother's first milk is rich in immunoglobulin A. And the antibodies are also in there and gives baby passive immunity for months. That is why the health personnel always advise that you give exclusive breast milk for 6 months so that the baby can get immunity to survive the environment. Passive immunity, we are looking at antibodies that are obtained from somebody else. So like the baby receiving that from the mother. But when the baby becomes sick, then the baby will get his or her own immunity. Okay? His or her own immunity. And because the mother is giving the baby that immunity, the baby does not know how to prepare the immunity. It's not able to, because it doesn't know how it was prepared. The mother prepared it and gave it to the baby. Okay, so it's borrowed antibodies. But when the baby becomes sick or the individual becomes sick, then the individual will manufacture the immunity himself. Now let's look at the disorders of the immune system. We are looking at immunodeficiency diseases. We have severe combined immunodeficiency disease. That picture is showing this young man stepping out, but you can see that no part of his body is exposed. Everywhere is covered because he has a hereditary lack of B and T cells, and he's vulnerable to all kinds of infections. Okay, all kinds of opportunistic infections. So because of that, he stays indoors all the time. If he has to step out, he has to wear this kind of protection, protecting his head, protecting his arms, protecting his legs, and the rest of his body. Right. The next one we are looking at is AIDS. It's standing for HIV. AIDS is a severe form of HIV. There is invasion of the helper T cells, and the macrophages and the dendritic cells are tricked and internalized. The viruses are the retrovirus. It uses a cunning way to destroy the helper T cells, because the helper T cell is the one that will go and tell the story. We watch a short video on HIV and AIDS. So this is just what happens to the virus. When the virus enters the body, it goes to the DNA to release its genetic material into the DNA and changes the enzymes that the original DNA has. And so the ribosomes in the cytoplasm will translate the RNA into the viral protein, not the individual's protein, but the virus protein. And HIV or AIDS is a disease that involves the destruction of the CD4 cells or the helper T cells, so that nobody will tell the story. Okay? Virus, HIV, not lack of self-antigen, that causes the AIDS. And it's a retrovirus. It uses the uses of antivirals only is the way that this virus is being treated. And why there is no vaccine available is because the virus keeps changing itself. Every time the virus changes itself. So right now, we have HIV type 1, type 2, type 3. So if you have it, or if you have somebody who has it, or a patient has it, the patient must go strictly according to the doctor's advice. Okay, because what is used to treat HIV type 1 is not the same as medication that will be used to treat HIV type 2, and so on and so forth. Okay, so this is how the HIV virus looks like. This is how the HIV virus looks like. So we are going to just look at a few pictures, okay? That is Kaposi sarcoma. Kaposi sarcoma is to do with something of the virus. And the HIV transmission is through blood, through semen, vaginal secretions, breast milk, and across the placental barrier. So because of that, retroviral mothers are not allowed to have normal vaginal birth. When the time comes for the baby to be delivered, it is delivered by cesarean section, and they are advised strongly not to give their breast milk to the babies because they will become infected. And since that has been done, a lot of babies have not gotten the HIV virus in their blood, and they have survived. They have survived the environment. Most common means of transmission is sexual intercourse, vaginal, anal, or oral, contaminated blood products, blood transfusion that is contaminated, using of contaminated needles, not transmitted by casual contact. Undamaged latex condom is an effective barrier for HIV. So if you want to have sex and you know you are HIV positive, you have to use the condom. Okay? You have to use the condom. Other autoimmune disease, you are looking at the immune system not recognizing its own self. It doesn't recognize its own self, but it destroys its own tissues. All right? Other example, we are looking at multiple sclerosis, where the white matter of the brain and the spinal cord are destroyed. Myasthenia gravis, which impairs communication between nerves and skeletal muscles. Juvenile diabetes, it destroys pancreatic beta cells that produce insulin. Rheumatoid arthritis destroys joints. Self-tolerance is to do with the T suppressor cells. They create surface antigens, which is a surface protein that makes all of your proteins recognized as a self. And this protein must be continuously made throughout your life. If you stop producing it, you get autoimmune disease. One of them is rheumatoid arthritis. So this is rheumatoid arthritis. It's an autoimmune disease. Autoimmune disease. The immune system does not recognize itself. It starts destroying your own body cells. Okay? And this is lupus. Lupus is also an autoimmune disease. Autoimmune disease. Okay? I want us to watch that video, and we bring the topic on lymphatic system and immune system to an end with this video.
Okay.
Okay, well, this seems like a good time for a snack break. Yeah, medical research is tough work. That's left to professionals. Dear Tim and Moby, what is AIDS? It sounds pretty scary. From RIS 106. AIDS is short for Acquired Immune Deficiency Syndrome. It's a disease that damages the body's immune system. Well, your immune system is your body's way of fighting off sickness and infection. T cells are an integral part of the immune system. They stand guard over your body waiting to organize and drive off invading pathogens. A virus called HIV, or human immunodeficiency virus, can cause particular trouble for your immune system. Instead of just trying to get into your body, it attacks the T cells directly. When enough T cells are destroyed, the immune system is weakened. Illnesses that your body can ordinarily fight off, like the cold or the flu, can get in and cause serious health problems. As you may have guessed, HIV is the virus that causes AIDS. A person is said to have AIDS when the immune system reaches a really low number of T cells and it gets to the point where the body can't fight off infections. Well, you don't catch HIV like you catch a cold. You can't get it from hugging, holding hands, sharing a sandwich, or sitting next to someone who has the virus. And it's not spread by mosquitoes or swimming pools. HIV is only spread when the bodily fluids from one person come in contact with the inside of another person's body. Most people contract HIV through unprotected sex or intravenous drug use, and the disease can pass from a mother to a fetus in the womb. Well, there's no cure yet, but progress has been made with treatment. People with HIV are living much longer lives now than they did 15 years ago. HIV is difficult for medicine to deal with because it's a sneaky little virus. Sometimes one treatment will work for a while, and then the virus will mutate or change form so that the medicines don't recognize it. Doctors and scientists are working hard on a vaccine to prevent AIDS, just like the ones we get to prevent measles or mumps. But there's still a lot of work to be done before a vaccine or a cure can be found. I'm not sure they'll let you back in the lab. I mean, you've already broken like 52 test tubes today. Hey, maybe you're just too clumsy for lab work.